A very good afternoon to one and all. Welcome back to the second session of today's talk. We have a national expert talk of the GYAN course by Dr. L11 Drug. And before starting the session, may I request Professor Yasha Hasija, Local Coordinator GYAN and Head of the Department of Biotechnology, to welcome Dr. L11 Drug, Group Leader ICG. Sir has obtained his PhD degree in Physical Chemistry in 2003 from Tata Institute of Fundamental Research, Cypher, Mumbai. During his PhD work, Sir worked on NMR spectroscopy of protein and nucleic acids. His work includes the discovery of a novel C-tetrad in DNA quadruples. Sir also worked as a postdoctoral fellow in the group of Professor Kurt Wuthrich at ETH Zurich, Switzerland. After that, Sir returned to India in 2008 and joined the International Center for Genetic Engineering and Biotechnology (ICGB), where he works as a group leader. Sir was instrumental in establishing the high-field NMR spectrometers at ICGB New Delhi and NII New Delhi. His group works in the area of protein nucleic acid recognition with special emphasis on understanding the specificity of the recognition during transcription. Sir is also an adjunct professor at the Department of Chemistry, Indian Institute of Science Education and Research, IISER Bhopal, Madhya Pradesh, and at the Special Center for System Medicine, SCSM, School for Sanskriti and Indic Studies, SSIS, Jawaharlal Nehru University, New Delhi, and so frequently writes science articles for the common public and has edited 2 books, Biomedical Imaging Instrumentation, Applications in Tissue, Cellular and Molecular Diagnostic 2021, and When Science Meets Spirituality 2024. With this, I request Sir to please deliver his lecture.
So thank you, Neha. It's nice to be here again. So I'll talk mostly on the, I'll talk mostly on the how the drug receptor interaction can be quantified. So as you see from here, so our group is actually involved in the most of the time with how we deal with the bases or nuclei and how it interacts with the protein. And then this is the protein molecule. And ICGB is an international organization under United Nations umbrella. It has 3 components, one in Trieste, where it's a headquarter, and then New Delhi, also India, and Cape Town in South Africa. There are 66 member countries all around the world. So let's move directly to the part. So what are drug discovery and what are the pain and bottlenecks? So there is, once you identify a target for a drug thing and drug in the market, so there is a long gap between that. And in order to bridge that, there is a long path actually. You have to build a bridge for that to bridge this gap, and it takes really a long time for that. So when you have to take the drug in the market. So target identification in drug market, one can go by different methods. So in this case, what you see, one can try different way of approaching the other end, taking a drug in the market, and in between there are many problems it encounters, and only few of them actually survives. Most of them are actually not surviving to the end, and it may be a possibility that the fastest one which you are thinking that it may go may die also in between. So one of them will reach. So what are the challenges? The challenges are, for the drugs, it has to be efficient and it has to be high throughput. That is a global challenge which we have, and especially with respect to India, it has to be affordable also. So this is a big thing which we have to keep in mind. It has to be affordable also. So in a drug discovery pathway, when you start, it's like that you start from a disease, then you have a target, then you have an assay, development, probe to lead. After that, it goes to preclinical studies, FDA, then phase 1, phase 2 trials, phase 3 trial, finally review, and it reaches clinic. So there is a long path, and normally this takes 10 to 15 years of time. So how to shorten this path? Normally, means a way is there that one can shorten the path by eliminating few of them. And what it can be eliminated, normally it is in the repurposing. You can get rid of preclinical FDA phase 1, phase 2, phase 3 trials. From here, once you can go directly. So this is the way in the repurposing of drugs. So, you have a reduced safety and pharmacokinetics uncertainty and using existing drug for new treatment. So, this is repurposing of drugs. So, strategy normally when you start this drug discovery part, you use experimental structure or model the structure of the pathogen protein and refine the structure again by energy minimization and molecular dynamic simulation, then screen with millions of compounds against the target protein from different libraries, select the common inhibitors based on different parameters like chirality, chemical integrity, solubility and all other things, and finally check for potency of the inhibitor using energy minimization and MD simulations. Select the best inhibitor, and after that the in silico work is over. You start with experimental, then in silico work, and finally what is a long path is experimental validation. So, in this experimental validation, the first step is the molecular interaction has to be quantified, and almost any chemical reaction or physical change is accompanied. So, when it is a binding, it is a change in heat or enthalpy, either it is absorbing the heat or releasing the heat, and which can be termed either exothermic or endothermic in nature. A measure of rate at which a heat exchange is taking place with the surrounding is simply equal to the rate of reaction. So, this is the rate of reaction is directly proportional to rate of heat exchange with the surrounding, and which we call the enthalpy. So, in a particular thing, if you see the receptor, upper equation, receptor binding to ligand, a forward rate can be quantified, is called Ka, and backward rate when the complex AB is breaking is called Kd, the dissociation rate. So, K equilibrium is again coming from the complex concentration, receptor concentration, ligand at the equilibrium, and it can be defined as a K capital Ka, Ka by Kd, and then this is directly inverse to the 1 by Kd dissociation constant. So, small k is the rate and bigger one are the equilibrium constants. And delta G simply is a log of K equilibrium multiplied by the R and the temperature at which reaction is occurring. So, delta G is the standard, and delta G at particular moment is actual concentration at particular time of complex, receptor and ligand plus the standard delta G at free energy of the molecule. So, this is a typical thermodynamics and kinetics of any interaction which is taking place. And this also can be directly quantified in that change in enthalpy minus temperature, change in entropy, that also gives the delta G. So, kinetics and affinity constant, one has to be clear on this. The rate constant describes kinetics, how fast molecule binds or dissociates from each other. Association rate constant, K is at which complex is formed. Dissociation rate constant, Kd, shows that at which complex is dissociating. Affinity constant describes the strength of reaction. So, this is small k and this is a capital K, strength of interaction. Proportion of complex and free interactants at equilibrium. Constants are characteristic for their interaction at particular solvent condition and physical condition like temperature, pH, ionic strength, etc. Constants are independent of the concentration of the molecule. So, if you look at the rate constant, association rate constant and dissociation rate constant. So, forward reaction A plus B forming AB, K dissociation, AB dissociating to K, and this is Kd. Units are generally molar inverse per second inverse, and this is second inverse. Meaning, its rate of complex formation, that is number of moles of AB forming per second in 1 molar solution of A and B, and this second inverse is the dissociation rate constant, stability of the complex, that is the fraction of complexes that decays per second. So, if your Kd is 0.01 second inverse, means 1% of complex decaying per second. Range of Ka is generally in 10 to the power 3 to the power 7, and Kd is inverse of 10 to the power minus 1, 10 to the power minus 6. Similarly, for equilibrium constant, equilibrium association constant and equilibrium dissociation constant. So, by definition, this is both are inverse of each other. One Ka, equilibrium association is molar and dissociation is molar inverse. association constant defines that binding strength, high K, high affinity for the complex formation, and dissociation KD defines dissociation tendency, high KD, low affinity. Range are generally are for affinity, association 1 10 to the power 5 to 10 to the power 12, and dissociation 10 to the power minus 5 to minus 12. Typical range in biological interaction. When we see the biological interaction, typical ranges are, you have starting from the rate constant going from 100 to almost million, that is very fast. Dissociation rate constant again inverse of that, so it is 1 by 100 to 1 by million, and affinity constant goes starting from weak, that is 10 to the power minus 3 to 10 to the power minus 15, which is the strongest one, the biotin and streptavidin, which are the, and in between somewhere the antibodies association comes, and then signaling molecules which are the weak because they have to associate and dissociate depending on the condition where the molecules are interacting. How to quantify these interactions? For quantifying, the macroscopic one are the classical ones. So if the best thing to understand how to quantify is that anything which can separate signature of the free one from the bound form can be used to characterize the interaction and quantify it, and the error in measurement of the free and bound will be directly going into the error of the quantitative characterization of the interaction. So the macroscopic ones are the classical one. You have the gel where you do a gel shift assays, or the absorption spectroscopy where you have the different signature of the free and bound, or the fluorescence where also you have a different signature, either intensities are changing or the peaks are also shifting. Kinetics way to quantify the interactions are surface plasmon resonance, or the BLI, which uses the magnetic interference, and using a thermodynamic parameter to characterize, which is the isothermal titration calorimetry. Microscopy, when you go to atom specific interactions, there are structural techniques, mostly the NMR spectroscopy, X-ray crystallography, and cryo-electron microscopy, which does the characterization. So macroscopic again, classical ones. So kinetics, SPR, you do again quantification. Now coming to the thermodynamics part. Thermodynamics, SPR, you have a kinetics because you have the one surface bound to the one molecule, other is flowing over that, and then you measure the rate constant, and using the rate constant you determine the affinities, interactions. In ITC, you actually try to put the molecule in the ligand to the protein and try to see the heat change, which is compensated again. So it measures the delta T between the free and the bound one, and then applies the current so that it again come back to the same temperature. So that temperature, and that's what it's in, and how much current is applied, you measure it, and that is the thermogram. Once you fit it, you have this kind of fitting, and from the saturation level where all molecules have saturated to the bottom gives the change in heat, the delta H, and it describes the mechanism. The slope of this one gives the affinity, one inverse of the dissociation constant, and in the middle of this line defines the stoichiometry of the interaction. Now you see why ITC is important, because here you see that there are two thermograms, and if I measure the rate, the binding constant, both are same, because these two lines are running parallel to each other and the slope of them is same. So the KD remains same. But what is different is here is the delta H change. So delta H means its enthalpy is different. So the mechanism is different. So now let's come to this part in a more clear manner. So there are three interactions plotted here, N, A, B, C. So now you see the red one is delta G. So delta G is same for all three, implying that the affinities are same. Now there are differences in delta H and delta S. So A is, if I look at A, is enthalpy driven, means it's a strong hydrogen bonding, van der Waals interaction coupled to a conformational change, because you see the delta S also is large. So there is a large conformational change also happening. B is entropically driven. So there is a large change in entropy, and then it is also enthalpy change. So it's a mostly hydrophobic interaction and possibly rigid body. While the third one is mildly enthalpic and mildly entropic. So a small negative or positive enthalpy, expulsion of structured water molecules from the binding. There is not large conformational change, and releasing this water molecule will increase the entropy. So now if I look for the drug one, the third one, the C, is the best one because it's not causing the large conformational change. It is specific and only release of water molecule. So that's why ITC becomes quite important in quantifying the interactions. By other techniques, you generally understand only the affinities. You don't understand the mechanism of the interaction. Now coming to the microscopic part. So there are techniques for structure determinations. So it's like if you pack the molecules, if all the molecules are packed and you want to look it. So in this case, these are like a solid, means it's a structure, it's a crystal, either the crystal like you grow the crystal of salt or sugar at home. Same thing, the protein molecules is also crystallized and packed in an ordered manner, and then you try to look how they look like. So by either X-ray crystallography, X-ray diffraction, neutron diffraction, cryo-electron microscopy, and also solid state NMR spectroscopy is also used for that purpose. So how it goes, that you have a concentrated protein solution here, and then you apply certain chemicals so that it comes out of the solvent and it crystallizes and forms a nice crystal. And once you have the crystals, let's assume these are the atoms for here in crystals, and you shine X-ray light on them, and because X-ray lights are shining and because of the electron cloud around each atom, it will diffract and produce the spot. Since these spots are coming because of the electron cloud, so in turns one can get information of the electron cloud around each atoms. And this was done by this father-son duo, Henry Bragg and Lawrence Bragg, who developed this technique of diffraction, X-ray diffraction, and producing these spots, and both of them got a Nobel Prize. So once you have these spots, because it is coming from the electron cloud, you can go back and do the Fourier transformation and calculate the electron cloud. This electron cloud now can be fitted to your molecules, the amino acid in this case, because you know the sequence, and you start fitting each of the atoms in this electron cloud. Once all electron density is satisfied, and including the water molecule somewhere in between, you get the structure of the molecule. So this is the way you get the three-dimensional high resolution structure of a biomolecule in this case. And first structure of the alpha helix was done by Lennox Pauling. Later Max Peruz and John Kendrew did the structure of hemoglobin and myoglobin. G. N. Ramchandran did the structure of collagen, the triple helix. And what was interesting, that Ramchandran made a conformational map of the 5' the protein when no structure was known. So he used the ball and stick model to have the 5' map, which we call now Ramchandran plot. And when we have more than 200,000 structures in a protein data bank, this Ramchandran map still holds true. So that is the beauty of his calculation, which he has done based on the steric hindrances. Looking at the electron density, so when we say the protein structure is solved at particular angstrom resolution, what it means. So when you say at 6 angstrom, you see a large density and you don't see the individual atoms. When you start going down, you start at 2 angstrom, you see more and more clarity resolution. 1.5 angstrom, you start seeing all the atoms, because single bond length are generally 1.54 angstrom. So at 1.5 angstrom, you start seeing all the atoms very clearly, and resolution become much and much better clear as you go down. So advantages of crystallography: it's a rapid technique, high resolution and precision, because you are packing and crystallizing and then you are looking at them. You can very clearly see water and ion interaction. Well-defined contacts for inhibitors are also seen. So you have a protein ligand interaction very well characterized, including the involvement of water molecules during the interaction. Limitations: it's just the one snapshot. So when you are crystallizing a molecule, it generally follows the symmetry rules. So it crystallizes in a one particular symmetry, and So other conformations are either shifted back to shifted to this particular equilibrium, or they are just outside of that. So you once see only one kind of conformation. Generally, these molecules, protein molecules, are live. Actually, they are living entity. They breathe, and they are multiple conformation also in the native states. Intrinsically disordered protein cannot be characterized by crystallography because they can't be crystallized. Many times one see missing electron density of flexible region because they are flexible, they have high B factors, so you can't actually get a structural information from those region or very poor region. Many times when the ligands are dynamic, one sees the poor density, and that way it becomes difficult to characterize. If one wants to screen multiple inhibitors, it's a slow screening process because all the time one has to crystallize with particular ligand and then solve the structure and look at this. Even for mutational analysis, it's a slow part because if you have to mutate any residue, mutate it, again crystallize it, solve the structure. In some cases, it may not crystallize at all if the mutations are there. So that's why it is slow. And when it comes to dynamics, it's again a very slow technique. It provides very, very limited information. Although B factor can in some way provide a dynamic information, but it's a very, very crude way to characterize the dynamics using crystallography. Cryo-electron microscopy, which has become very popular in recent days, in this actually one doesn't crystallize the molecules. So you actually, biomolecules are embedded in a vitrified ice, and then 2D projection are taken of electron micrograph, and then this is used to determine the orientation and origin of each particle, and finally you have a 3D construction. So millions of images are actually acquired because molecules are in this vitrified ice embedded in different orientation, and if you take a 2D image, then different direction these images are actually coming up, and once you have the millions of images, 3D construction can be easily done. So this is the simple principle of cryo-electron microscopy. So this is what is one molecule is oriented like this, other is like that. So you have a 2D images of each of them like that, and similarly, so for so many orientation, if you acquire images, then one can construct back this 3D image of a molecule. So this is the way one gets the structure in a cryo-electron microscopy. Next comes the solution part. So if you want to really look the molecules as if they are swimming around, not the packed fishes, packed molecules. So if you want to look at like that as if they are living ones, there is a technique which is called solution state NMR spectroscopy. You can look this molecule. So this is more like if you are looking at a still photograph and a videograph in this case. So you are looking at a different conformation lively. And there is also a technique which has come up in recent. There are only few facilities in the world right now, X-ray free electron laser, where the nanocrystals are used. So what happened in NMR of proteins? So in NMR of proteins, each hydrogen atoms of a molecule gives a resonance absorption, different absorption spectrum, because they are in different chemical environments. And since they are in different chemical environment, they are different electron cloud, and because of different electron cloud, they see different magnetic field. And because they see different magnetic field, they produces different absorption lines. So it is directly a signature of what kind of secondary, tertiary or quaternary structure it is having. So this is a typical spectrum, proton spectrum of a protein molecule. So you see all of these lines are absorption lines are coming from all the hydrogen atoms present in this protein molecule. Typically, all the aliphatic ones, the methyl, delta, gamma, beta, and alpha comes upfield of the protea, this water line, and then you have the amino group, aromatic group, and the amino group.
So how are we characterizing these hydrogens in alpha, beta, gamma, delta?
Characterizing means?
So like what is H alpha, H beta, H gamma?
So these are the alpha, H alpha, you have the amino acids, alpha protein. So these are the proteins.
Okay, in that sense.
Yeah. So generally these are made of alpha amino acids, so alpha protons or beta protons, then side chains I'm talking.
Side chains. Okay, these are representing
Side chains. So side chains are generally seen here. And aromatics comes between around 6 to 7 ppm, NH2 comes here, and all the NH group comes here. So there is another technique which is a two-dimensional spectrum. So this is which gives the fingerprint. What it does actually, in this case, you have the magnetization coming from proton to nitrogen. then it gets the chemicals information from nitrogen, and again it goes back to hydrogen, and then you detect. So you get the chemical shift of both the hydrogen and nitrogen. In this case, this is like a polypeptide chain. So for a polypeptide chain, you have in one axis you have a nitrogen chemical shift, the absorption lines, and in this other axis you have the proton chemical shifts. And if you cut a plane out of this 3D, you get this kind of spectrum. So this is called a 2D HSQC spectrum. So you simply cut a plane from this cube and you get this kind of spectrum. So this is called 2D N15 HSQC spectrum of a particular protein molecule, where each line, in this case coming from each N15H present in this protein, and normally you have all the backbone NH is one for each amino acid except for the proline and the first residue. So if you have the 4 residue here, so we will get 3 peaks. You will get clearly 3 peaks, each coming from one HN group. So means it signifies that number of amino acid present and number of the peak present in the 2D N15 HSQC spectrum. So in this case, a very clearly 22 residue peptide is there. If you have a 22 residue peptide, there are 3 prolines. So 3 prolines will not give any lines because there are no NH. First one, tryptophan, will not give because it's a NH3 plus group. So if a 22 minus 4, it should have 18 lines, 18 peaks. So 18 peaks you can see here clearly. And then you have a tryptophan side chain here because indole ring also has a NH group. So that comes, tryptophan side chain indole group comes around 10 ppm. You have a one Q here, and Q has a one NH2 group also. Q have one NH2 group here also. So this NH2 group, because one N is connected to 2H, so that's why you see on one N line 2 protons here. While this one is coming from backbone, this is coming from the glutamine and asparagine side chains, and here it comes the tryptophan side chain. For a 100 residue protein, this is a kind of a spectrum you see, where each peak here is for backbone NH, and these paired ones are coming from the glutamine and asparagines. Another characteristic way to identify the side chain ones, because you see a nice cap on the peaks. This is coming because of the isotope effects, and they will be in pair. So one can easily identify these NH2 groups coming from that. The advantage of NMR spectroscopy, you get a dynamic structure of a protein. Further, it can easily characterize intrinsically disordered protein because you don't have to crystallize the molecule, and you can get all the information from that. Easy to study flexible regions. Dynamic ligands can be studied also in this case, because if there is no fixed thing that I'm looking at that, so even the molecules which are ligands are dynamic can be easily studied. Rapid screening of inhibitors is possible in this case. So you have, once you have a protein molecule solution, then one inhibitor is bound, then second time you can have second inhibitor, and then you can easily do. Mutational analysis is fast in this case. Sorry, it's a typo error. It's a fast in this case. Mutational analysis is fast in this case because you have to mutate and record the N15 HSQC spectrum and understand it. And then you have a dynamics. The NMR is the best technique to characterize the dynamics, ranging from the picosecond motion to the second motion. So that way NMR can easily characterize dynamics of a molecule. Limitation, it's a slow technique as far as the getting the structure is concerned. It takes time. It has a size limitation in the liquid state NMR spectroscopy. It's difficult to go beyond something like 80 and 90 kilodalton to get a complete structure. All the structure informations or different active sites people have derived even from the Groyles complex, proteasome, which are as large as 900 kilodalton and 20s per. NMR structures are generally low resolution and precision as compared to X-ray crystallography. We don't get information on water and ion interaction because we don't see. If we have to see the water, there are other techniques, but we get a bulk information from that. Limited information on contacts between macromolecules and inhibitors. So now coming how we characterize in NMR the binding, same thing, the receptor ligand interaction. So let's assume that my protein M binds to a ligand here and it forms again ML complex. So if I say the signature coming from M is A, and M when it is bound to L, the signature is B. So what happens? So when it is very slow. So NMR time scales are in millisecond. So if it is very slow compared to the NMR time scale, then we will see at equilibrium A, the characteristic chemical shift of A coming here, and characteristic chemical shift of M in the complex, which I call B, coming here. If it is very fast compared to NMR time scale, I will see a very average peak here. And in case the exchange between this is in the time scale of NMR spectroscopy, microsecond to millisecond, I will not see any resonance here. So this is again used to characterize the interaction. So now you see if the chemical shift difference between A and B is delta, and fast I called if the exchange is faster than 2 pi delta, slow is slower than 2 pi delta, and intermediate, as I say, it is exchange is comparable to 2 pi delta. So this exchange regime can be changed either by altering the temperature. So if I raise the temperature, this exchange will become very fast. So it will go from here to here. So one can look at, so if in case somebody is not able to visualize the resonance line here, one can go from here to here very far. One can go from here to here very far by changing the temperature. So this is again in N15 HSQC. So what you see here, then the free one, this is the resonance peak of each amino acid. As I put my ligand, it's slowly changing from here to here. So as it's like you putting the molar equivalents. So 0.2, I can put 0.2, 0.4, 0.6, 0.8, and then I go to 1 and 1.2. So if the stoichiometry is 1:1, after that nothing will change. So it is slowly going from from the free to the bound peak direction, and this is a fast exchange regime. So NMR is able not only to characterize this thing, but it's saying how fast the ligand and a protein molecule is exchanging with each other in the bound and free form. This is a 2D HSQC spectrum in a slow exchange. So slow exchange, as I said, one will see both the peaks. So in the free, I am seeing only one peak. As soon as I start titrating, I start seeing this peak in a free and the bound state. Slowly it is growing for the bound one, and once completely the whole molecule is in the bound state, I will see the peak coming from the bound peak only. So this is in the slow exchange regime. So one can characterize what regime it is following. How to quantify this interaction? So this is this we had the RNA, this is from our group, RNA protein interaction. So we have the protein spectra, and slowly it is getting titrated. One see few of the peak are shifting, some are not shifting. It means those amino acids are actually involved in interactions, and that's what you see here in the zoomed region of particular area, that it is, and some are actually not changing at all. So these are potentially are not perturbed by the interaction, and those which are changing are perturbed by the interaction. So if you look at the free and complex, the two endpoints of the titration, so I get this kind of spectrum. And what you do generally, you calculate the distance between the free and bound form by applying simply this Pythagoras theorem, and chemical shift in N15 dimension divided by 5 and proton dimension, take the square and root of that, so that everything is in positive, because some peak are shifting in both the negative direction and positive direction. So make everything positive, you square and take root, and this 5 is coming from the empirical region, because the spectral width of nitrogen is almost 5 times compared to the proton. So that's why you divide by 5. And once you have a distance between the free and bound form, you can plot this as a function of residues. So now I have plotted the changes as a function of residue, and the peak which are showing the maximum changes are the ones which are getting perturbed by the interaction. They may be directly or indirectly getting affected. So it means their chemical shift are changing. Some may be remote because if the large structure change has taken place, and some are directly. So if these regions are potential. So now if the structure, one can do with the carbon 13 HSQC. So if you want to look at the site change, so how they are changing. So this is the site chain HSQC. So one can look at which are my gamma, delta or the methyl groups are changing upon binding to the ligand. So one can look at 13C HSQC also. Once when we have this thing, so we know these are the residues which are getting perturbed upon binding, and we can map this on the structure of the molecule. Once we map on the structure of the molecule, we know which these are the regions. So in this case, these are contiguous region in this protein, and that's why this is binding to the my RNA molecule. So even before characterizing or determining the structure of the ligand bound state, I could get the information of where the molecule is binding, and that's why it becomes quite easy to do a different ligand binding before determining the structure where the ligand is binding. So that's a rapid screening of the ligands one can do in this case. So, and one can also get a structure, kind of a structure by docking. So this is not a de novo docking. This is the docking with the restraint which I got, the experimental restraint. One can use the program like Haddock to get a bound structure. So in this case, this protein binding to the nucleic acid using this chemical information, we got this is the way it is nicely binding the protein, the RNA interaction. One can do an image analysis. So this is an area of image analysis. One can simply do an image analysis to understand how different they are. So this is again using machine learning or AI part. Transfer NOE, when the one molecule is quite huge and other is small, like one is characterizing tubulin or something, and how it is binding to a drug like Taxol or something. So this is technique it is used called transfer NOE. When the KD is greater than 10 to the power minus 7 molar, when there is a fast exchange, and one can standard collect the 2D Nogier spectrum. So these are three parameters has to be satisfied for observing the transfer NOE. So transferring ligand shows a single set of resonance average over bound and free form. So it is again because it's a fast exchange, so it again shows the average over bound and free form. Ligand is generally kept 10 to 50 times access related to the protein. A strong NOE developed in a complex is transferred to the free ligand state and measured from the free ligand resonance. Applicable to large molecular weight complexes because we are looking only at a ligand signature. Observed NOE can be used to determine a bound conformation for the ligand and change in the signs of NOE cross peak related to the diagonal. So this is the one spectrum that this is my ligand and this is my large macromolecule, it is binding. So this is the off rate and this is further going into the unfolded state. So one gets a Nogier spectrum before addition. So you are looking at the Nogier spectrum of this molecule ligand before this macromolecule is added, and you get this peak. Once after this macromolecule is added, so you see a cross peak coming up. So this is a cross peak, and this is what we call, it's a very small percentage of receptor I am adding in this case, and this has taking the information from the macromolecule. So one can get this information. Another technique is saturation transfer difference, NMR, a simple and fast method, but people have developed. It has robustness, focused on the signals of ligand. No NMR information of the receptor is required, and in a small quantity of non-labeled macromolecule can be used in this case. It is called saturation transfer NMR. What it does here, that you have require a spectrum of off resonance spectrum, on resonance spectrum, and then you calculate the difference. So difference, if it is you are seeing intensity of both, can provide the information how it is binding to the macromolecule.
So one question, in this transfer anyway, the ligand was 10-50 times associated to protein. What is the reason?
So because you are observing only ligand, you want to observe the ligand concentration, and only a fraction of ligand should be bound on that, so that it takes the information from the macromolecule. That is the reason.
So what if we do it like equal quantity? What will happen then?
So then everything will be bound state. Then you will not get the up signal from that. So I want it to be come off. It has to bound and it has to come off. And while coming off, it gets the information from the bound state. If it is not coming off, then I will lose that part. I will not be able to see the resonance line from that ligand.
So that information that it is taking is like the conformation change that it is.
No, it takes the, when it is bound, because of the Nogier transform from that, NOE transfer, because short distance, it takes this information and comes out. So during the time you get this information. So So this is the magnetization transfer. So magnetization is transfer from the macromolecule to the ligand.
Okay, it's a physical feature.
It's a-
No other.
No.
We study like mRNA transfer.
No. It directly is a magnet because of the distance. So generally it's a outer power 6 dependence. So if it comes close to the macromolecule, this will get transferred from the macromolecules to the ligand, and then it is coming off. So once it's coming off, it has this information from the ligand micromolecule. See, this is again the example of few drugs which saturation transfer spectrum has been obtained. So if this kind of residual peak one sees, then clearly it is getting bound to the ligand. So the different technique, you have a CSP, you get a shift, you have the Loxy experiment when you get completely peak reversed. T2CPMG, you have the binder here, and non-binder you don't see any change. Binder you see a intensity minus target, it is like this, and plus target it is reduced intensity. Saturation transfer, we have already seen that you see a difference. That's all. When you have no binder, you don't see a difference.
So what are these things?
The different spectroscopy. So chemical shift, perturbation, which I showed, peaks are shifting. So that's what you see, peaks are shifting in this case. When you don't have a ligand, peaks are not shifting. Binder,
W-Loxy, what is it?
it's a water experiment. So water you get experiments. It's another spectroscopy.
Okay.
These are different experiments. Different spectroscopy you use to characterize different kind of interactions.
You're getting different results.
Different. So you do the magnetization transfer different ways, and then you get a different kind of spectrum. Depending on what kind of ligands and what kind of molecules you are using, you generally select which one you have to use. Most of the time people use CSP because it's easy to do. So people can also use drug screening inside the cell. So they are called in-cell NMR spectroscopy. So you can use the signal from the protein molecules in the living cell. So this is one paper which people have published, that looking at the inhibitors of MTB growth by in-cell NMR spectroscopy. Because you see, again you are looking at the molecules as if swimming. So directly you have the cells, MTB cells, you can directly take the MTB cells in NMR tube and then characterize it. And look at if you put the drugs, if your drug is going inside, either by different saturation transfer or different technique, one can get the information from these things. So these are few examples of the in-cell NMR. You see the difference peak here. Now intermolecular NOEs. So these are the ultimate thing to characterize the final structure of the bond complex. In this, what one does actually, you have the one of the molecules, generally the protein molecule, is labeled with stable isotopes. So these are stable isotopes, N15 nitrogen or N15 nitrogen and carbon 13, because carbon 12 is non-NMR active and 14 has just 3 lines per, it's because it's a plus 1 spin. So generally half spin is 15N. So you label by growing the protein in the labeled minimal media, where you solely supply 13C labeled glucose and 15N ammonium chloride, and so that all the molecules are actually incorporating these nuclei. And finally you purify your protein, which is uniformly labeled with N15, if you give only N15, or if you give both, then N15 and 13C. And then your inhibitor is unlabeled, you are not labeling. What you do here, that you try to see how close they are coming by looking at intermolecular NOEs. And in this case, what is the different techniques are used, that you have a 13C labeled molecule, one of them which I said, and other is 12C, then you see the cross peak. So if it is a filter editor experiment, it means it will have the NOE peak only from 13C level proton to a 12C level proton. And if you have a filter nosy, you will see the NOE peak only from the 12C attached protons. So these two techniques are used to characterize the complex. So this is one of the spectrum of a protein binding to nucleic acid, where the protein is labeled and nucleic acid is not labeled. So in the filter experiment, I will see only the cross peak, which are the hydrogen atom attached to 12C. So this is a typical RNA spectrum. I am not seeing any peak coming out from the 13C label. So one can also have a 13C separated nosy. So you have a nosy when you have everything, and then you have a filter separator. So in this case, the peaks are coming because of these atoms are close to the protein molecule. These are to the protein.
So can you go back So, in this one you said, I guess C13 representation is missing.
Huh.
So why?
So we are, because carbon 12 is NMR non-active and 13C NMR active. So I apply a pulses so such that 13C attached protons. So what happens when you have a coupling constant? The carbon 13 proton coupling constant is roughly around 140 to 160 hertz. So when I apply a pulse, because there is a coupling constant there, and other 12C attached proton, because 12C is not NMR active, there is no coupling constant. So I apply a certain pulses so that the peak attached because of the coupling constant completely is washed out. And because these are not attached to the C13, they are not coupling constant, so they will be visible.
And this is the coupling constant for C12.
C13, only NMR active nuclei can have a coupling constant. 13C attached 1H. So I am applying that those which have a coupling completely get washed out, which are not applied. So this is again from the three-dimensional spectrum. So you can have many peaks in 13C because the protein-protein interacts. In protein, many protons are coming close to each other. But when you have a 13C filtered and separated, you see only the cross peak between the ligand and the protein molecules. So once you have all the information from the distances, so this is a thing. You have a noisy, very crowded one because it's showing all the proton-proton connection in the protein. But in this case, it is showing only the protein thing which are close to the ligand. So only those peaks are seen, ligand peaks close to protein. And once you have this intensity information, one can calculate the structure of a molecule in the bound state. So in this case, we had calculated the structure of a RNA bound to protein because we have all the distance information, most of distance information, which proton is close to which proton of the protein, which RNA proton is close to which proton of the protein molecule. So using that distance, I can fix the distance. I have the distance information, I can fix it, and then I allow it to solve it. So I get the information of the complex. So this is the way you get a complete structure of the protein bound to the ligand. One can use this thing to determine the KD also. Of course, NMR is not the best technique or the ideal technique to determine KD, but in many cases when other techniques are having problems, then one can use NMR spectroscopy to determine the binding constant of a ligand to the protein also. So this is one of the structure we had solved of the Plasmodium SR1 structure bound to the RNA. This is bound to the RNA, and in this case we wanted to see the which residue is contributing to the binding of the RNA, which are the residue having what affinity contribution. So in this case, what he had found on from the structure different conformation, that which in how many this ensemble, which are the one actually bound to the thing. And in this case, what we see, there are only 3 nucleotides are close to that. So this is actually providing the real affinity for the interactions. See that. So this is in case in the ensemble, most of the time it is seen, and C5 and A6 we are not seeing at all. So this is, that's why one can say these are 3 residues, 3 nucleotides which are necessary for the recognition of nucleic acid to the protein. We can also look at the individual changes in intensity as we titrate, and we can plot it to see which residue is providing how much contribution to the global affinity. So residue specific affinity can be also calculated using NMR spectroscopy in this case. So this was another case which we have the protein structure bound to a DNA, and it has a very unique binding. So it's a unique packing one. What was seeing that molecule had packed, sandwiching the nucleic acid between the 2 molecules. And when we had done ITC and other things, we found that the stoichiometry is 1:1. While what in crystal packing we are seeing 2:1, that 2 molecules are binding to the 1 nucleic acid. And nucleic acid was very well seen. So there was no problem with the electron density. So we could easily see an average B factor was 78.7. So we can see each and every nucleotide very clearly in the crystal structure. So there are different ways. So first thing, different structures are deposited based on the which experimental method was used. So in case if it is somebody has solved with crystallography, somebody has solved with NMR spectroscopy, so there will be 2 structures. two different structure. Third thing, today I have done a protein structure by crystallography. I got a resolution up to 3.2 angstrom. Next day, some other lab tried to do something, refine it, and got a higher resolution at something like 2.2 angstrom. So then it will be a different ID. And third, like if I have a protein molecule free, I have solved it, then I mutate it, then again I solved it, then both will have a different ID. Third part, if I have protein, then protein is free, I have solved it, somebody else or I myself solved it with a ligand bound, then inhibitor bound or alternate ligand bound, alternate inhibitor bound. So all will have different IDs. So there are, how many times the structures are solved, each will have separate ID. So in India, we also have now at 6 places we have high resolution cryo-EM in India, 6 places. So these were supported by Sathi Foundation, Sathi scheme of DST. So in Delhi, the IIT Delhi has for all the Delhi. So one can use cryo-EM, but cryo-EM is growing because now you can look the bigger complex. So like in SARS-CoV, if I have to characterize a spike protein, a spike protein is a trimeric structure. And it's a huge one. It has a plasticity. So I don't want only RBD. I just want to look whole thing. So people have used the cryo-EM to get a complex structure also. Many times means many mutants could not be crystallized. So then the cryo-EM also is the best way. You don't need a crystal 3D packing. You can do with a 2D packing.
So these mutants are different from the wild type, right?
Yeah. So that was in the COVID. That spike was changing a lot.
State-wise data we were having.
Yeah. So its sequencing data was there, and if people are able to isolate that virus or recombinantly produce that protein, you from the sequence information mutate it and produce it and then solve the structures. So that's why when you are looking, and that's why I showed initially, when you are looking at the structures, always pay attention few of the things. That always resolution is a very important thing if you are doing crystallography. So or then second part is which are the residues for which B factors are very high. B factor high means those residues are not properly defined. They are dynamic, they are flexible. So one has to be very careful if you are commenting on the biological function or saying that okay, this is my state, this is not the correct state, it may be something flexible, very flexible. One has to be careful on that. If you are looking at NMR structures, one has to see where the NOEs are highest. Restraint file. If you see a bundle, like in this case, if I go back, okay. If I see this area, it means it is not compact. So this area is dynamic. This area is dynamic. This
area is not compact, it's dynamic.
Dynamic, yeah. So it will. If you look at the experimental data, it will have the less number of NOEs. So it is not. It is poorly defined structure. It's a poorly. While this area, this is very well defined. Now you see generally what is expected, and loops have to be dancing. Loops should be dancing, and that's what we see. Loops are kind of a dancing here, and all the C terminal and N terminal ends are also dancing. They are freely dancing. And well, the rigid part, the global part is well packed. So NOE, we have to see how many NOEs they have.
What are NOEs by the way?
Nuclear overhauser restraint. So nuclear overhauser restraint means the magnetization from one atom can go to second atom using the dipole-dipole coupling. So magnetic dipole coupling. So because these are magnetic dipole, each atom nucleus has a magnetic dipole. So magnetic dipole goes to there, and this Hamiltonian is R to the power cube dependence.
It's a Hamiltonian operator.
So Hamiltonian, so it's a dipolar Hamiltonian. So what we had, and it has angular dependence also, but in solvent it is freely dancing, so one can assume to be that part angular dependence is gone completely. So, and this is R to the power cube dependence, and for the intensity it will obviously be the square of that. So it has a R to the power 6 dependence. Okay, so that's why if it is two atoms are close within a 5 angstrom, then magnetization can get transformed from one atom to the second atom. So then you see the cross peak. Less than 5 angstrom. So that's why you see a cross peak. So if I have a 2 peak here, and if I see a peak here, off diagonal peak, it means these two atoms are close in space. Close in space.
The magnetic dipole coupling can be transferred.
So because it can be transferred, that's why I'm seeing the peak.
Okay.
So I do a Noji spectrum.
Okay.
Noisy spectroscopy means I allow the magnetization transfer to happen. In that technique, I allow magnetization transfer to happen from one atom to another atom. It will get transferred to only those atoms which are close in 5 angstrom speed. So it will not get transferred to all other atoms. So that's why I get a distance information.
So the C13 example that you were telling, so when you did this, the C13s were all washed away because
Yes.
it was in vicinity.
No, that was a J coupling, not a dipole coupling.
Okay, fine.
So there are two things. It's a J coupling is a scalar coupling. It's a true bond.
It's a vector coupling, right?
Yeah, and dipole is a vector coupling.
It's a vector.
It's a pure vector. And J is a scalar
J
coupling.
is a scalar coupling.
J is a scalar coupling. So because it is true bond, it transfers true bonds. It's a linear thing, a scalar coupling. Okay. But this is transferred with the distance and angle, r theta dependence. So it's a vector.
Right, so
Dipole is a vector. So in this case, so in this case, when we saw that, now so we have this unique kind of a packing, crystal packing. And from all other data, we knew the stoichiometry is 1:1. In solution, we have to confirm this thing, because this is just a crystallization artifact, because it's the symmetry which is followed, it packed in this direction. So we did the same CSP calculation in this case. So we have the labeled protein molecule and we titrated with the DNA molecule. And what we saw, some of the peak are shifting. And once we had this peak shifts, we plotted this peak shift as a function of sequence. And what we saw actually, that what we see in the crystallography is same in the solution, but with the one molecule of protein only. So it means that what I saw here, that DNA is talking to one RRM1 of one molecule, and this part is talking to RRM2 for another molecule. Actually, in solution, what is happening, that this is talking to only one molecule of that. It was just a crystallization artifact, which we can resolve using the solution technique, because solution technique is the actual technique which is happening inside the cell area. Crystallization can happen because it can get biased towards particular symmetry, a space group. So we confirm that this is binding in this manner, and this is the final way that, okay, what structure we have solved from the crystallography and what NMR are consistent with each other for interaction. Dynamics, one can look, as I said before, it can characterize dynamics starting from the second to picosecond, femtosecond scale, like diffusion one can quantify, easily chemical exchange can quantify, molecular rotation, vibration, all these things can be quantified by dynamics information obtained from the NMR spectroscopy line widths. So this is a classic example of HIV protease. So HIV protease is an aspartate protease. What we see, this is an active site where aspartate is there, and when it is get resistant to different inhibitors, you see mutations coming at different places, which is not in the active sites. So the HIV molecule is able to evolve its molecule without compromising its activity by changing the residue somewhere here there. So which is different. So it's a kind of allosteric effects. So how to do that? So then people are doing crystallography also. Then they found what is the role of this, because we see a lot of mutation happening in this region, the flap region or the hinge region. This is happening. Nothing in the active site.
So what's the cause to this? What may be the cause to this?
Means the nature actually is evolving to keep plasticity. Means I'll show in the next slide, which you understand. It doesn't want to compromise its activity. So HIV protease wants to remain as active as it is, but it wants to throw out the inhibitor very fast, so that its own survival is not at stake. So how it does actually?
It's like an evolutionary mechanism.
Yeah. So this is the way HIV molecule happen. So this is the active site, and this is the flap region.
The active one.
Yeah, the flap region, flap. So this is the flap region. So when the flap region is moving very fast, and if the molecule can actually change some residue. It can make either fast or slow to allow the ejection of that one particular inhibitor which you are targeting. And in that way, it allows the conformational plasticity and a lot of conformation. And the one which doesn't allow inhibitor to efficiently remain inside the active site, it will be selected by that. So this is the way nature actually evolves.
Like a natural selection.
Nature, yeah. So naturally it evolving. That's why it evolves, and that is the reason you see the mutation. And this dynamics again, this motions here and this region, his region. You see the motions in this region. You see the motion in this region, and that's what you see. The mutations coming up in these regions. So this is the, so viruses are generally smart, quite smart, without having brain. So this is the way it is being used, and these informations are generally used for the structure based design of any drug. Recently, AlphaFold has come up, and it is a big way. It's a machine learning tool and AI tool, and it is evolving. And AlphaFold 3 has actually evolved to a certain extent that it is able to give the structure of molecules at very high accuracy, especially for the monomeric. There are problems still with the multimeric proteins where the buried surface area is less dimer, or the domain exchange dimers where it is also having a problem, but it also has a limitation as far the intrinsically disordered proteins are concerned. So where AlphaFold you can't use, because it's again it's a disordered protein, nothing can happen, and everything is actually built because it's again a learning process. So it is built on the whatever structure protein data bank has. So it has learned from that, learning has taken place, then it has evolved and it has gone into detail after that. Now we have many of the proteome completely solved from using AlphaFold 3. I'm solving and I'm repository. It's my primary database, and I'm giving the structural coordinates. I'm giving the XYZ coordinate of each atom. So it's a primary database. But people may think, okay, because you have a sequence from information from somewhere and it is coming from that, it is a secondary information that it is sequence set is mapped to the structural map set. But I personally, as a structural biology, I think it's a primary. Let's assume a very thing. I make a hypothetical protein. I make a hypothetical protein, and there is absolutely no sequence map present from the downloaded one. And I determine the XYZ coordinate of that and deposits. So it is purely my primary. So one can employ AlphaFold, but again, it is again a word of caution. For many times, biology means generally those who are not working in computational biology or structural biology, they think everything is solved. No, it has learned from the available database. And let's assume, means most of the folds people have identified. Let's assume that there is a novel fold. Then this will not. Then this will not.
Or maybe the feeding database have some inherent biases.
Biases. So, yeah. So this can be a problem, or something is, means let's assume, I give you a very classic example of prion proteins, A beta proteins. So prion proteins, there are places where it changes from the alpha to beta completely, and then it aggregates. A beta, it's a helical structure which completely changes to beta, and then it stacks over each other, forms amylide, and finally fibers. It stacks. So same sequence space is giving to my two different structural conformations. Same thing, alpha synuclein, it aggregates. So those places, so one has to use it. It is good for many things, but again, it's not like with a blind eyes or blind thing. And to see, and then you again try to understand, but you apply the brain to see whether it is really doing correctly or not. Whether
Lab validation is required, I think.
Yeah, many times wet lab validation will be required. But let's assume, means I have a protein which is something like, say, monomeric protein from human and from a mouse. Somebody has determined from mouse, and it is a monomeric protein having a similarity of almost like 50-60%, and they are made. So it can be very accurate. It will be as accurate as a crystallography. So these things one has to look into this. What structure I am predicting. So that's why. But AlphaFold, the second information which one miss a lot is the water molecules. How water molecules are interacting with different residue. they forms a very good interaction thing, because many times the ligand interaction with the protein is mediated through water molecules. So water molecules will not be predicted, and water molecules comes many times the crystallography also how it packs out the different water molecules, but people look at the conserved water molecules, the conserved molecules, and whether if I am putting the ligands, they are playing a role in the interaction or not. Maybe hydrogen bond is mediated through a water molecule. Means I will say it's not much of physics has gone into this.
So that's what physics,
Is
energy,
physics has not gone into this. It's just takes tetrapeptide range or peptide, what structure is there in different one, what it could have, and then predict it.
So conformation wise, it's nice.
It's something it uses kind of a distance geometry, you can say,
Yes, sir.
kind of a distance geometry. But yeah, that's why many times you have to use AlphaFold. You run an energy minimization and at least do a 200, 300 nanosecond MD on that. At least perform a 200 MD on that. Then you try to use it for many other purposes. So that is where I really thank